Welcome, everybody. We're very excited to have Professor Lee Miller here today to tell us a bit about development of a universal biomimetic computer interface. Are we decoding from a tortured manifold? Professor Miller is coming to us from Northwestern University in the Neuroscience, Physical Medicine, and Rehabilitation, as well as Bioengineering department. So this is going to be, I think, really exciting. So please take it away.
All right, thank you. Good morning. I'm glad to be here. I've literally been here maybe 3 or 4 times at this point, and I'm still getting over the view that you folks have. It's awfully impressive. What I'd like to tell you about is work that began 30 years ago in my monkey lab that I ran literally across the street, across a couple of streets here at the medical school campus, which I've subsequently closed. But my initial work with monkeys was to investigate how the neuromuscular system controls our movements of our arms and hands. And that was basic research for at least 2 decades. And then I began to get involved in what the field calls brain-computer interfaces. We did that for a while with monkeys, and now I'm working with humans in a collaboration with the University of Chicago and the University of Pittsburgh, humans who have tetraplegia due to spinal cord injury, and trying to translate some of the things that we did in the monkeys to the humans. So there's a bit of a clinical aspect to this, clinical in quotes, potential translation in the future, but I've added a few slides at the end that are a bit more mathematical in nature, which are the result of fairly recent work that we've been doing to try to understand this thing that I'm referring to as a potentially tortured manifold. So I'm very curious to see what you folks think of this and what ideas you may have. The basic outline of the talk is this. First of all, what is a biomimetic intracortical brain-machine interface? I hope some of you already know this. I'm going to put some fairly basic slides at the beginning to make sure that we can all come along with the basic talk. What's a neural manifold? That's sort of the central part of this talk in some sense. How can we encode decoding across many tasks? The decoding refers to looking at the brain and decoding a signal related to the monkey or to the person's intent to move that we can do something with. And finally, how tortured is this manifold, this mysterious manifold that I've been referring to? So let me get right to the essence of the story, and that is what happens when a person has spinal cord injury. Now, again, many of you know this already. I suspect perhaps all of you do, but the nerves coming off the spinal cord come off at different levels of the spinal cord. So I'm sure you've all heard about spinal cord injury at different levels. A low-level spinal cord injury that happens, somewhere down here affects just the lower limbs, the legs. But a high-level spinal cord injury up in the cervical spinal cord leaves a person with tetraplegia, quadriplegia, weakness or paralysis of all 4 limbs. So depending on that level, the severity of the injury varies. Now, basically what's happened, of course, is you just disconnected the control signals that originate in the brain that can no longer get to the muscles. So there's a lot of interest in developing these brain-computer interfaces which record those control signals in the brain and use them to affect movement, because those signals no longer get to the muscles. But the brain, the motor areas of the brain, is still intact. It's not quite normal, because the other thing that happens in spinal cord injury that I'm not going to talk about is that the ascending signals from the spinal cord are also interrupted. So in addition to being paralyzed, you lose the sense of touch and the sense of proprioception. So in that respect, the control signals, the feedback signals that the brain normally gets are not normal. So the system is somewhere between that in your brains and someone who's had a stroke, where the cortical circuitry is clearly disrupted. Here we have an intact cortical circuitry. It's just not getting the inputs that it normally gets. This is just a little bit of neuroanatomy. The whole brain here drawn in cartoon form. I don't think you can see my pointer, so I'll just refer to it. There's a premotor area. There's a somatosensory area. I'm going to walk over here. I'm walking out of the camera, I realize. This is what's called the central sulcus, which divides the motor part of the brain from the somatosensory part of the brain. So it's that little wedge in there, the motor cortex that I'm going to be talking about. And on the right side, you can see just a functional diagram of how it's connected to the spinal cord. It goes through interneurons in the spinal cord and then to motor neurons, which are the neurons that actually cause muscles to be activated. So basically, I'll be talking about this relationship between primary motor cortex and muscle activity. I want to pause, however, and make one more basic kind of comment. For those of you who have never heard a neuron, you can stick an electrode into the nervous tissue, into the brain, into the cortex is what I'll be showing you. These neurons that you can see, they're the little black blobs. A monster big neuron is about 50 microns across. They're more typically 10 to 20 microns. So we put a little microelectrode close to the neurons, and I'll show you in a moment how we do this in the context of a brain-computer interface. These neurons signal electrically, and I'm not going to go into the details of the channels and the ionic flow and such, but basically there's a small change in potential on the order of millivolts, tens of millivolts, which accompanies what we call an action potential. Cells talk to one another, or they talk to muscles by emitting these action potentials, and those are the deflections on that oscilloscope trace that you see at the bottom. They last for about a millisecond, and they encode information in terms of the firing rate of these action potentials. So you can look at it on an oscilloscope, you can collect it on a computer, you can also listen to it on a speaker. This was a video that I took a number of years ago in my lab. This monkey's been trained to do a variety of different motor behaviors, but in this case, it's very simple. He's going to reach out and grab some raisins out of the wells in this thing that's called a clover board. And I want you to listen, because the sound that you're going to be hearing is going to sound like noise. It's the clicks of these action potentials from a single neuron in the motor cortex. So you should be able to correlate the sound you're hearing with what the monkey's doing and get some sense of what this neuron may be involved in the monkey's motor behavior. I'll cut right to the chase. It's a hand area neuron. So as he was beginning to reach, this neuron became quite active as the hand was opening. It's easy to confuse that with the movement of the arm, which is highly correlated with it. And as we study this system, these kinds of correlations among the various movement correlate signals are problematic, because if I hadn't told you, many of you would have quite likely have assumed that this is a neuron controlling reaching as opposed to hand movements. So there are a variety of ways in which we can tease these relationships apart, including fairly complex mathematical approaches, which we do in order to try to study the system in a basic way, but we also use related kinds of algorithms, of approaches, to compute what I'm going to refer to as a decoder, and I'll show you that in just a moment, which takes the firing rate of not just one neuron, but many neurons recorded simultaneously, and computes an algorithm relating them to a control signal that we'd like to produce and use for restoring movement. This, in fact, is a slide of what we call the Utah array, and it's pictured up in the upper left. It's 4 millimeters on a side. It's got a 10 by 10 grid of electrodes, which are each 1.5 millimeters long. And you can see on the right, this is a picture during surgery with one of my monkeys. We take this array, place it on a part of the cortex that we would like to record from, and there's a fancy pneumatic inserter that punches it down into the cortex. The electrodes are 1.5 millimeters long because at that depth within the cortex are found the neurons that project down to the spinal cord, and they carry the output signal from the cortex. So in the monkey and in the human, in which we use exactly the same kind of electrode array, we'll place them like this, we'll screw those pedestals to the skull, and then put the bone flap back in, close the scalp around it. When the monkey recovers from this surgery, he's got these 2 connectors coming out of his scalp, which we can plug our instrumentation into and get recordings like those on the bottom. So you think about those as a whole bunch of little oscilloscope screens showing action potentials of the sort that you just heard. There are different colors of action potentials, because if you think about this electrode tip and these 20 micron neurons around it, some of them by chance are going to be close to the tip, others farther away. That's going to change the size of these action potentials for sure, and also their shape to a certain extent. So the computer can look at the different potentials that it's seeing on a given electrode and cluster them into different shapes. That's the basis of these different colors. So essentially, we've got 96 electrodes are actually wired to those connectors in one of these arrays. If we have a really good implant, we can record 2 neurons from each electrode. So occasionally I've had monkeys in which we're able to monitor 200, 250 neurons simultaneously. That's not always the case. Often it's less than 100. But in any case, many tens of neurons that we can record from simultaneously of the sort that you just heard. And these are carrying different kinds of information, some related to the hand, some related to the arm. And by looking at that aggregate kind of information, we can study the brain or we can use these as control signals. I'm going to pause for just a moment. I was going to say at the outset, if you have questions as I'm going, please feel free to ask.
What cortical layers are your electrodes typically in, do you think?
So I referred to the output layer, which is layer 5 in the cortex. In a monkey, it's about 1.5 millimeters down. So we're probably recording from layer 5. It's the cortex is a bit curved. The array is flat, so they're probably not all in 5. In some cases, they may not any of them be in 5, depending on how well the insertion went.
What fraction of layer 5 are you recording from?
Minuscule. I showed that the cartoon that I showed you at the beginning was a human brain. The primary motor cortex is, I don't know, 2 centimeters across perhaps, and 4 or 5 centimeters long. And I told you this is a 4x4 millimeter array. So it's a few percentage of the primary motor cortex, let alone the rest. This becomes an important point. I also bragged about recording 100 or 200 neurons simultaneously. There are 1 million neurons going to my arms to let me move my arms. So it's a huge undersampling that we're doing as you know, I'm talking about these control signals that we're producing. Okay, great. But Need, what can we do with them? I referred in passing to using exactly the same array that I showed you in humans. And in fact, this is Jan. She's a quadriplegic, and she was implanted a number of years ago at the University of Pittsburgh. And you can see coming out of her scalp, just like my monkeys, this connection to these pedestals. I'm going to start this video in just a moment, but she's controlling this largely anthropomorphic robotic limb, which was produced with funding from DARPA in the Revolutionizing Prosthetics proposal. That arm is about $100 million arm, not each one, but the development cost for it. It basically duplicates all the degrees of freedom of the human arm. She's not controlling all of them. She's controlling 7 degrees of freedom. So she can position the hand, she can change its orientation, both in 3 DOFs, and she can open and close the endpoint. And this is just some indication of her progression in performance as she practiced this thing, which she started out being able to control the thing in 3 dimensions on day 1, but it took a number of months before she could do what I'm going to show you here. So she's reaching out, orienting, grasping this block, and moving it over. And remember, she's doing this without the normal tactile feedback or proprioceptive feedback. So part of her slowness and clumsiness is due to the relatively poor motor control that we're able to provide her, but part of it is due that she doesn't have the normal feedback either. But nonetheless, she's doing a remarkably good job of grasping these different objects, moving them, even manipulating them to a certain extent, driven by roughly 100 of these million neurons that you and I would use. Yeah.
Does she have access to does she have visual?
She has visual feedback. That's the only kind of feedback. If she didn't have that, it would be hopeless. And they've done a variety of experiments, indeed, removing visual feedback, moving her arm, and so forth. Any other questions? So that was a number of years ago. It's no longer quite the state of the art, but in fact, the state of the art continues to be the work done with respect to robotic control at the University of Pittsburgh and Chicago. This was 7 DOF. I think I have an example of 11 DOF or 10 DOFs where they give the hand a number of additional degrees of freedom as well. Yeah.
Does she have any kind of proprioception?
She does. And I, I realized as I made the comment about moving her arm, that didn't make any sense, but I had told you before. To digress just slightly, she has a neurological disorder that shouldn't have paralyzed her. It's related to the cerebellum, but she was paralyzed. So this was actually an issue when they initially presented these results. But in this respect, her feedback from her own arm has been preserved. So some of the experiments they did was to move her own arm in synchrony with the robot arm to try to give her that feedback. So there's a lot of stuff like that that they've done. It's great fun to talk about, but I can't possibly get into here. Okay, but what I do want to point out is, I think I mentioned they're controlling the velocity of rotation of these different DOFs or translation. This is something your motor cortex did not evolve to do over millions of years. It evolved to control muscles. So one of the words in my outline was that I wanted to develop a biomimetic brain-computer interface. So that grew out of my studies over a number of years, looking at the relationship between motor cortical activity and muscle activity. So the basis for the BCI that we developed in my lab, and that has been used by other means in other labs and clinically, uses what's called functional electrical stimulation. I had a slide back from 400 years ago where they discovered that muscles were electrically excitable by, getting a spark of electricity to move them, but I decided I didn't have the time to show that. But this couple of clips comes from the Cleveland FES Center at Case Western Reserve. This person has an injury at about C4, C5, so he's quadriplegic. He can move his shoulder. He has no control of his hand. I don't know exactly what he can do with his elbow, but that all depends on the level of this injury and the particular nature of it. He has stimulating electrodes implanted on a bunch of the muscles of his forearm and the intrinsic muscles of his hand, those that allow you to move your hand. He also has sensors on his shoulder. So when he moves his shoulder, there's an external controller that has been pre-programmed specifically for him, such that when he raises his hand, it stimulates the muscles in the right spatial temporal sequence to cause his hand to close. So if you watch on the left, he's raised his shoulder, closes his hand, stays closed, or dropped his shoulder and his hand opens. This is the basis for what they call the free hand prosthesis. And the other clip on the right shows another patient using this thing, and it's pre and post-operatively. You can see rather dramatic improvement, and you see him shrugging his shoulders in order to pick this stuff up. It allows him to do this box and blocks paradigm significantly better than without. He's picking up a pen and writing with it as well. There was something on the order of 200 patients implanted with this prosthesis over, I think, a couple decades, one in any case. Many of them had this kind of improvement, but the thing was withdrawn from the market because it wasn't marketable, basically. So it's no longer available. But we're using this kind of technology in our monkeys, except instead of having it pre-programmed, we're trying to control it by our recordings from the brain. And if you think about it, what these people are doing successfully is literally using the same muscles to position their hand as they're using to open and close their hand. So there's got to be some amount of cognitive burden, and clearly there's going to be motor learning involved in learning to use this thing. We'd like to essentially wire the brain to the muscles in a way that makes that process easier to learn. This is a monkey sitting in my lab, and you see the same connector thing coming out of his head. It's not actually connected at the moment. We trained him to do a task that was inspired by this box and blocks, essentially. He'd pick up the ball, put it in the tube, it rolls down, and he does it over and over. The figure on the right shows the recordings that we made from the brain on the top and from muscles down below as he grabbed this ball 3 times. And you can see the bright colors in the neural recordings. You see the deflections of EMG, the electromyograms of several muscles involved in the task on the bottom. There's a variety from reach to a certain extent because he does it a bit differently, and certainly different muscles have different patterns of activation. But what we can do is look at the firing rate of these neurons, and in this case, simply do a simple multiple input linear regression between the neural activity and each of these muscles. A little more specific, this is an impulse response, Wiener filter, many different terms refer to it. Some of you will recognize them. But basically, we look back in time 500 milliseconds, which we divide into 10 bins for each of these neurons. So we have, in this case, 70 neurons, about 700 regressors that we fit to the activity level of these three different muscles. This gives us this linear mapping, this decoder from the brain to muscle activity, which we can cross-validate by collecting new data and seeing how well predictions using that algorithm, that decoder, match what we actually record. And that's what's shown here. So the blue is the predictions, and the red, I think I have it right, is the actual recordings. For the most part, they lay over each other quite well. We computed the R squared between these two signals as a measure of the accuracy of our predictions, which ranges from a low of about 40% up to 80% for this muscle. So this becomes the real-time control signals that we're going to use to stimulate muscles. Yeah.
You said you use just linear regression?
This is just linear regression.
So is that a surprise or is that not a surprise?
No, because the brain controls muscles. If you knew me, you know why I'm smiling as I give you that answer. I promoted this idea that what motor cortex does is controls muscles. It doesn't control joint rotation or a hand position, that sort of thing. So the fact that it's approximately linear was not a huge surprise. It's only approximately linear, and in fact, that's a lot of what I want to talk about. It's time varying, it's complicated. And we can do better with a nonlinear map, for sure. Yeah.
Earlier that there's a lot of undersampling in these.
Yeah.
Like, can you quantify what you mean by that? Or
well, I gave you a couple of numbers, which in one sense quantifies it. We record 100 neurons, there are 1 million that are controlling it. So do the math. It's tiny undersampling. It's not quite that bad. And in fact, that's where this manifold comes in. And this is what I'll get to in just a moment.
As you add neurons to your regressor, does the R squared improve?
Yes.
But it must.
It saturates. Yeah.
Okay.
So we don't know exactly where it saturates because it doesn't saturate. Certainly it begins to saturate at 100. It also depends how complicated the thing you're trying to get the monkey to do.
What are the other 999,000 neurons?
Excellent question. We may come to lunch. Come to lunch. There's all kinds of interesting questions like that. Potentially they're denoised. So these are noisy signals. So one of the things we're doing is just averaging across a bunch of neurons instead of over time. I hope I get through this, but I like to talk about interesting stuff that you find interesting. So I told you we use 50 millisecond bins. That's a fairly noisy estimate of the firing rate of these neurons. We could record for 500 milliseconds and get a much better estimate, but now our time resolution goes to pot. Alternatively, we could put them in 5 millisecond bins. That becomes basically a binary process because neurons don't fire that fast. But if we can record average across many, many neurons, then we can go to 5 or 2 millisecond bins, get really good temporal resolution and good noise as well. That's part of why there's so many. It's certainly not all of it. So what I haven't told you is, well, I haven't told you the people that did that experiment 15 years ago. Emily was a student, Chris a postdoc. They worked out the stuff that I told you about the decoder part of it, but they also developed a system in which they put cannulas around the nerves that go through the muscles, through the arm to the muscles, which were connected to a subdermal injection port right here. All that means is they could take a syringe and make a single injection into this port, and the local anesthetic would go to the nerves and paralyze the nerves for a couple of hours, like when you go to the dentist and get Novocaine in your jaw. So that left the monkey's hand paralyzed for a couple of hours, during which time they could do this experiment. So they recorded from the brain, they made these predictions, they stimulated muscles that had been paralyzed. So if it works, the monkey can voluntarily modulate his motor cortex, and his muscles should move accordingly, contract accordingly. This is a video of one of those experiments. The first couple of times he's reaching, the paralysis is in place, but the prosthesis has not yet been turned on. So it shows you the deficit. His proximal arm is completely normal because the block is below the elbow. So he can reach out, but he can't grab the wall, even though he's trying. And presumably he's modulating his motor cortex. So now it comes on in the next couple of trials. These are intermittent trials within the experiment. If it's on, he can grasp nicely. If it's off, he can't. And he can coordinate this completely natural, normal shoulder muscle activity with the the computer electrical stimulator produced movement of his hand. Okay. We had, I think, 5 electrodes in 3 muscles in the hand. The hand has something like 40 muscles. So the monkey could not do very much with his hand. He couldn't do this because we couldn't activate the muscles to do that. But he could open his hand. He could, squeeze a little harder on this side than that side. And he could modulate how hard he was squeezing as well, all voluntarily. And I have a little bit more evidence of that claim here. In addition to the task I just showed you, we trained him to squeeze a force sensor in what we call a power grasp. And he had to squeeze to different amounts to put the force, which is the white thing that he was producing, into these target windows. You see, on the 4th one, he failed completely, and that's because that was one of those trials where we turned the FES off. We could continue to make the predictions shown in red. He was trying, and in fact, he tried twice, because the first time the force didn't go up, he stopped trying, then he tried again, the second red pulse there. And then waited for the next target to appear. We turned the FES back on, and he got back into that target. Okay, so he's voluntarily controlling the amount of force in time to put this cursor into the window. I showed that mostly to show you this result. When you do FES, I'm not going to go into the biophysics, but it causes your muscles to fatigue much more than normal. This is a problem in our experiment. It's a problem when FES is used clinically as well. And you see that blue curve going down the left shows this rather dramatic decline in stimulus effectiveness that happened within the 90 minutes or so of our experiment. So we have to deal with that somehow. The monkey needs to deal with that. Remember, I showed you he was going into these 3 different target levels as he was squeezing this power grasp device. And yet I'm showing you here he's gotten 35%, he's lost 65% of his strength. So how is it that he's able to do those high-level force targets just as well at the end of the experiment as he was doing at the beginning? That's what's shown here with these bars. And the answer is he was simply trying harder. So if you look at the pulse width that was being delivered to those now weakened muscles, it got about 3 times bigger. That pulse width is determined by us looking at his brain. The algorithm says he's trying harder, so you have to stimulate harder. And it allowed him to produce the same force despite the fatigue. So it's another nice example that he's using this in a fairly natural kind of way, consistent with the idea that this is a more biomimetic sort of interface. All right. Neural manifold. How many of you are already familiar with this? Thoroughly familiar with this concept? Okay, a few of you, not all. So these little bars up there, I borrowed this from John Cunningham and Byron Yu, represent firing these single action potentials from 3 different neurons. Obviously, this is a cartoon. They proposed, or at least they made this cartoon after a number of people proposed, that you could take these firing rates and plot them, in this case, in a 3-dimensional neural state space. So each axis of this 3-dimensional coordinate system is the firing rate of one of these neurons. So you can locate you can locate the state of the brain or these 3 neurons at any point within this coordinate space. And as time goes on, you measure different action potentials, different combinations. You can sweep out a trajectory through this space, as shown here. Now, the important thing to get back to your question is that these data don't fill this 3-dimensional space. In fact, they lie on this cartoon on a 2-dimensional manifold. But this illustrates rather well what does happen in the brain. There are various estimates about the dimensionality of this real neural manifold. It's clearly related to what the monkey's doing, may be related to how many neurons we're able to record. There are lots of things that make it a little fuzzy, but it's on the order of 10-dimensional, probably. So the undersampling, if we can figure out this, if we can look at enough neurons to calculate this underlying manifold, the undersampling problem maybe isn't as bad as it first appears. This is another clip that came from recordings in my lab a number of years ago that I really like to illustrate this concept. These are now 40 neurons that were recorded from the somatosensory cortex. This is the part of the brain that supplies signals to the brain about how the arm is moved. So we had the monkey grabbing the handle of a robot, and instead of he moving the robot would bump him in these 4 different directions. And we measured the firing rate of these neurons in a little window after each of these bumps. So this produces a 40-dimensional vector or a point in this neural state space. Then we did factor analysis on this 40-dimensional space. And when we plotted the first 2 factors against each other, we found these points. Each of these points is one of the bumps of the monkey's arm. And the color of the point corresponds to the color, or to the direction of that bump. So you can see there are these clusters within this neural state space that's essentially isomorphic to the way the monkey's arm was bumped. So with just 2 dimensions, we're capturing quite a lot of static information about the state of this manifold, if you will. But we can also do a little video of the latent signals that we can calculate within this manifold that are related, we think, this is sensory related, but we can do the same thing in motor cortex, to the monkey's motor intent. So again, each of these dots is a single bump. I'm going to be showing you 100 of them at the same time in this clip. So this manifold, the activity is just kind of milling about randomly. It contracts for some reason just before the bump, and then you see them fly apart, and at the peak, they look like the thing on the left. So there's a lot of very useful information in this neural manifold, in this case about sensory information about the arm, but if we do the same thing in motor cortex, we can recover information about the monkey's motor intent in this manifold. And I'm looking at 2 dimensions here. I told you it's maybe 10-dimensional, so we can do stuff in 10 dimensions, but I can't show you all that. Okay? This is Scott. Scott is one of the participants at the University of Chicago who has a high-level spinal cord injury. He has tetraplegia. It's partial. And he's being plugged in just the way the previous participant I showed you was. And these are recordings as he attempts to move his thumb on the top and his index finger on the bottom. Don't know how many neurons there were here, but you can see the different patterns of firing rate across these different neurons, and that they are different when he imagines moving his thumb compared to when he imagines moving his finger. Now we do the same thing, project these into a 3-dimensional state space, and you can see the clusters related to each of his imagined finger movements. So you can imagine that there's a lot of information about the hand movements that he's trying to make that's available in this relatively small number of neurons. So we're hoping to use this as a means for doing these BCIs in humans. But there's a problem here. I didn't say explicitly, but I showed you in the monkey, we compute our decoder by having the monkey do something and recording his muscle activity and recording his brain at the same time. We obviously can't do that with Scott because he can't move. So people have done what's called observation decoding, which basically means if you want to decode arm movements, you show the person the movement he's supposed to imagine. He imagines doing it, you record it from his brain, and now you can compute the decoder between his brain activity and the motion that he was supposed to make. But we're trying to do muscle activity. And there are, as I told you, about 40 muscles in the hand. I can't possibly show the hand muscle activity to Scott and ask him to duplicate that. So instead, what we're doing is recording from able-bodied people, a bunch of EMGs, as they do a whole series of different hand postures, mostly taken from the ASL alphabet. Now we have the pattern of activity that normally occurs when you make these hand movements. We can ask Scott to try to make the same set of postures. And then since they're both watching in synchrony these same postures, we can get EMG activity from the able-bodied people, cortical activity from Scott, and assuming they're doing the same thing, we should be able to compute decoders on that basis. between two different people. Fabio did this experiment. These are the ASL gestures and some other stuff at the top. They don't line up with the EMG below, but it's those series of postures that contributed those modulated EMG signals. And again, to cut to the chase, this is how good our EMG predictions were based on Scott's attempts to make these same postures. They're not as good as in the monkey. There are a variety of reasons why that would be the case that I'm not going to go into. But you can see there is a pretty good correspondence between these quite a number of muscles and the recorded from the able-bodied person and our prediction of those EMG patterns from Scott's motor cortex. So I'm going to leave the human studies here and go on to talk about some of the dilemmas that we have to get back to the question of, is this system really linear? If there are no other questions.
Yes, there's no EMG from Scott.
Correct.
And but for this able like a subject, so there's no M1 recording.
Correct.
So
So they both need to be doing the same thing. So that lack of synchrony is part of the reason that our predictions are not as good. We're also assuming that Scott's musculoskeletal system is essentially the same as the person that did these, and that's a pretty good assumption, although we're investigating that question as well. Yeah.
It might not relate, but if I did something very similar with somebody that was amputee,
Yeah.
do you think you would get the same response?
Very likely, yes. With respect to amputees, there's an ongoing debate about whether we should use a brain-computer interface or a nerve interface, and a lot of people are working on that as well. There's a lot of overlap. Okay. How do we get more complex movements to study? These guys built this plastic cage. I'm going to start moving a little faster through some of this stuff. The monkey didn't live there, but he could spend hours there, and he would do spontaneous things like scratch himself, and you can see the modulation of wirelessly recorded EMG and wirelessly recorded M1 at the top. And then I missed it, but he grabbed something with a precision grasp. Now he walked across these bars. Now he's doing this power grasp at the left side. So this cage gives us the opportunity to get video, streamed M1 and EMG activity during a range of different behaviors that the monkey produces. This is yet one more two-dimensional projection of this neural state space. This time, it's for continuously recorded data while the monkey's in this cage. And this time, it's a UMAP projection rather than linear, for what that's worth. Schwen highlighted these three behaviors that I mentioned very briefly as I went through them. A power grasp of a device, a very similar power grasp as the monkey's walking across these bars, and then a precision grasp as he's taking food. And if you identify periods of this within this cloud of those three different behaviors, you see that when he's doing the power grasp, whoops, it ends up here. And then I was supposed to say, and it looks like that could be a trajectory. If you blow that up and connect the dots, you see that every time he does this power grasp, the neural state space follows this trajectory for a bunch of repeated trials. Same thing when he does the precision grasp over on the left in that purple, NU purple. And when he does the perch bar grasp separately from this power bar grasp, there's a similar trajectory that he goes through, but it's distinct from that. Each of these behaviors is distinct from the others. And all this gray stuff is unidentified behaviors. Monkey's scratching himself or doing who knows what. Okay? So this is our basis set to play with as we look at the brain. Yeah.
So you said here you used UMAP. So
Yes.
previously you used PCA?
Yes.
And,
Factor analysis was the one I showed you, but yeah.
and what are the differences? Why did you decide to use UMAP?
Well, UMAP, of course, is nonlinear, and that's the reason for doing it. It tends to pull these different behaviors apart more distinctly than PCA does. And unfortunately, I don't have time to go into the differences, but that's essentially what it does. It's kind of remarkable the extent to which it separates these different behaviors as clusters within the space. But PCA does the same sort of thing with different properties. Yeah.
Try a 3D UML.
I believe the answer is yes. It becomes computationally more challenging, and especially when things about going to 10. But yes, we have done some things in a higher dimension, more so in PCA when we want to go to higher dimensions than UMAP. Data sparsity also becomes an issue. So the first question is, how well can we predict these various different behaviors that we record from the cage? And the answer is very well. So power grasp, perch bar grasp, precision grasp are all predicted with very good fidelity across all of these muscles. But the catch here is these are single decoders that were trained in one behavior and tested in the same behavior. If we try to apply those behaviors, those decoders across behavior, they fail catastrophically. And this is just a demonstration of that more quantitatively. The diagonal is training on one behavior, testing on that same behavior. The off diagonals, there's all combinations of wrong decoder. They're all negative VAF. It's slightly better than that because if we train a decoder on all the behaviors, it does reasonably well on all the behaviors. Yeah. Okay, I went quickly through that, but I'm going to continue to go quickly here if there aren't any questions. So what occurred to Schwen is that any one of these clusters corresponding to individual behaviors, for the most part, can be decoded quite accurately with a specific decoder. So he came up with a piecewise linear decoder, all unsupervised. Basically, he identified clusters within this neural state space, not based on behavior, except to the extent that they were related to behaviors. And for each of those clusters, he computed a separate decoder. And now when he collected new data, he'd look at the data point, ask which cluster it belonged to, and use the appropriate decoder to decode it. Okay. This is an example of how this thing works. Again, for about 20 seconds of continuously recorded data. You can see the changing colors. These are the automatically changing cluster decoders that he used to decode this particular example. Again, I'm going to be going fast here, but stop me if you'd like to, because this is the punchline. That I showed you before. This is what you get if you train a Wiener filter on all the behaviors. This is what you get if you train an LSTM on all the behaviors, and you can see it's much better than the linear decoder. Surprisingly to us, when he had more than about 6 clusters, this piecewise linear approach outperformed slightly the LSTM. Okay. So by looking at these different areas within the neural state space and applying individual decoders, it works quite well. Yeah.
Did you think about applying a model with some kind of like switching dynamics, since you have these kind of...
Well, that's essentially what this does, unless I'm missing your point. Each one of these decoders has different dynamics between cortex and EMG.
I guess with the switching dynamical systems models, like you have a probability distribution that predicts the probability of your next state. So there's a kind of like temporal coherence across behavioral clusters.
No, we have not done that. Each of these points in the clusters is static. So except that we're using a Wiener filter, which adds some dynamics there, no, we're not doing anything like that. We still have a problem, though. If we leave out one behavior, again, that left out behavior fails catastrophically. Negative variance accounted for. So why is that? Fabio and Schwen both tried to pursue this question systematically, and this is the rather more recent kinds of data. So here's another point cloud. Now we're going back to a reaching task, and in fact, this is what we call random target task. And the whole point is to try to get a more uniform distribution of reach directions represented within this cloud. And I'm not showing you those reach directions, but there are directions within this cloud that correspond to movement in different directions. What these guys did was essentially tile this cloud with a bunch of holes that they punched out, and they computed the left out decoding accuracy within each of those holes. Just to see, I told you that leaving out a behavior fails catastrophically. What if you just punch out a region within this neural state space? How well can we decode then? And the answer is not bad, but you see there's clearly a pattern across these punched out holes in terms of which can be decoded well and which can't. So they punch out a bunch of different size holes as well as positions throughout this state space. And this is one of the summary plots that comes from this example. So the color is the size of the hole they punched out. X-axis is the left-in accuracy. So this is using all the data to predict the data within this hole. Left out is using everything except the left out to predict that same hole. So they're predicting the same thing with slightly different data. And what you should be able to appreciate is there's clearly a difference between left in and left out accuracy. Left in accuracy is higher, as you'd obviously expect, but not that much higher. And it does get, the difference becomes greater for the larger hole sizes, which you see in the yellow colors here. But basically, the left in accuracy predicts pretty well the left out accuracy. Other way around here. Okay, and I can also, so summarize one more aspect here. If you compute the distance from the centroid of this cluster, both the left in and left out accuracy fall off. So as you get out to the perimeter, you can't predict as well, again, as you might well expect. But there's not really that much difference between left in and left out, even for the big holes. So this, seems to contrast between this catastrophic failure of leaving out single behaviors and the reasonably good behavior of leaving out even a rather large hole that covers most of the whole space down in the lower right-hand side. So we're still kind of scratching our heads here, but we do know in some work that I will not present here, that adding in just a little bit of the left out behavior increases VAF significantly. So by leaving out the entire behavior, it's catastrophic. Putting in 10% of it restores much of our ability to predict. So it seems like there's something special about behaviors as opposed to areas, but we're still trying to sort that one through.
Have
Yeah.
you done an analysis to ask, like for each behavior, how many of the neurons in your set are really contributing to determining that particular behavior?
The answer is most all neurons for most all behaviors. It's just that their relationship changes. I can't give you quantitative numbers for that, but that's an observation many people have made. These signals are distributed across all of these neurons quite broadly. Of course, not uniformly distributed in terms of projection strength.
So is how well things work kind of proportional to how many neurons you leave in? Or, I mean, would you get better and better if you could make bigger arrays?
We've never left, well, yes, someone else, or maybe it was you, asked about the saturation. There's no question of that. Whether leaving out neurons that are more strongly related to particular behaviors makes more difference, we haven't tested. I assume that would be the case. But that remains to be seen for sure. Okay, last thing I want to show you is to try to get some sense of the shape of this manifold. We've been looking at these flat projections so far. This is the one you've seen a bunch of already. This Fabio did about 3 days ago. He divided this thing into a grid, and within each grid, he computed a decoder. Okay. Now this decoder, as I've been talking so far, would be 96 dimensional, its parameters. Actually, what he did was compute, do PCA, and then use the first 10 PCs for the decoder. So this is a 10-dimensional decoder. He did that for data sparsity reasons. But we have decoders of 13 muscles. So this vector that I meant to show you here is a 10-dimensional vector for each of the muscles that we're trying to predict. And he just averaged all that stuff together. So that's the direction cartoony for the decoder in that particular bin. Now he goes over to the blue bin, finds a bit of a different angle, different vector direction, goes to the pink bin, even more different, okay? So what he can do then is calculate the angle between each of these estimated decoder directions. It's a highly theoretical sort of direction, but it tells you something about the nature of the parameters in each of these decoders and how they vary as a function of the location throughout this state space. So to be clear, this is not a vector describing the orientation of the manifold itself, but it's the relationship of the manifold activity to the stuff we're trying to predict and asking how quickly does that change as you move across the manifold. This is the first bit of an answer. So in this case, what he's doing is calculating these angles, these vectors, and then he calculates 4 angles for all the cardinal directions around the center bin and averages those. So there's lots and lots of averaging here. But as you move across the manifold, it's fairly flat. It gets a little more abrupt changes at the edges, but not much at all. But those are fairly big angles. Those are 60 degrees, 70 degrees as you move from grid to grid. Okay, so this is sort of the local shape. This is what happens if you anchor the thing instead in the center and ask, how does that angle change as you move away from the center? And as you might expect, it gets more and more, the angle gets larger and larger as you move from the center, but not precipitously so. And in fact, as you get to the end, you get pretty close to 90 degrees. But remember, each of those bins on the left is about 60 degrees. So one of the dilemmas we have in interpreting this is that there are at least 2 sources for this angle. One is the manifold, the mapping is changing. That's the one we'd like to be able to find. But the other one is, to the extent that these decoders are noisy, they're just going to contribute noise. And within each of those small bins, he has only a few seconds of data to compute a decoder. So it's a noisy decoder. So you can see as the decoder accuracy goes up, the angle goes down. Okay, so to a certain extent, as you get more confident about this decoder, these angles get smaller. We don't know at all really how much noise makes up these 70 degree angles. There could be a lot of noise in there, and the variation from step to step is smaller. And in fact, the fact that the one on the right with the central anchor point doesn't change any more than 9 degrees suggests there's a lot of noise there in the left. And in the right too, for that matter. But the take home is there's nothing like an abrupt valley somewhere or obvious clusters related to the different behaviors that would lead us to believe that this is a really tortured manifold. It really looks as though it falls off fairly smoothly. And in some sense, that should make our efforts more easier to develop these decoders that span this large space.
Like in the previous slides, like in some grids, like there's different colors. Like for example, the first some grid has both orange and purple color. So maybe that could smooth out this kind of
undoubtedly, absolutely undoubtedly. So we played with the grid size. With a bigger grid, you're going to get more of that kind of mixing. With a smaller grid, you may get less of that mixing, but on the other hand, then your data becomes super sparse for computing the decoder. So we're between a rock and a hard place in that respect. We'd like to get really fine grid, but we don't have enough data to do it.
Why don't you vary your grid size across the domain as necessary?
Not keep a uniform size. Yeah. Part of the dilemma is where we have most of our points is where the monkey's sitting there doing nothing. It's uninteresting anyway. But we've not tried that. Certainly we could. Actually, sorry, I shouldn't say that. Well, no, we did not vary the grid size, but the number of points in each grid did vary. We ended up subsampling so that we had uniform points throughout the grid. So essentially all of them became noisy, but at least they all had the same limited number of inputs. This was literally 2 days ago. So this is where I'd love to get some feedback from you guys, sort of where to go next. But where I'm going to go next is my last summary slide. I told you about an iBCI. You can ask a question as soon as I get through this.
Okay, good.
And the concept of a neural manifold and the fact that we can calculate this maybe 10-dimensional thing based on recordings of about 100 neurons is really very fascinating and also useful. And we can find signals in there related to the monkey's motor intent. And as I just told you, we've got this manifold, and it looks as though it's fairly gently curved, although certainly curved. And I've shown you the folks who did most of this work already, and these are a lot of other collaborators and funding sources. And I also need to acknowledge the Cortical Bionics Research Group. This is the collaboration with the University of Chicago and Pitt that makes all of the human work possible, which I didn't talk that much about, but it's a critical component. So, your question.
Yeah, I don't know about you have geometry, like I don't know how interpretable that geometry is. I'm wondering if you have calculated, instead of like collapsing that into a low dimensional space, have you tried to just compute those angles in higher dimensional spaces and see whether...
Well, we're computing the angles, to be clear, in a 10-dimensional PCA space, essentially.
I thought it was in the human. Okay.
So it's a little confusing in that sense. You're looking at a 2-dimensional space, and indeed, for the experiments that I showed you, these are actually PCA. I just went back to the thing that you're familiar with here. But this is just a 2-dimensional PCA, and it's moving across the 2 dimensions, the 2 primary dimensions of the neural state space. Then we're calculating this vector in this much higher dimensional space related to the output muscles and to the parameters of those 10 PCs. I should also say, this is, as it says, PC1 versus PC2. If we do 3 versus 4, we get more or less the same kind of picture. 3 to 4, a little bit, but you have to squint. At that time, we don't have much variance left. But the story remains fairly true through about PC4. So any questions are fair game at this point.
So I'm curious whether you have a sense where sort of the other information about muscle activity might be. Right. So, you can do reasonably well from M1. And I'm also curious, actually, whether there's much of a difference if you have, recordings in M1 and S1, if you can do a bit better. But are there ideas about whether there are, other brain regions that, if possible, to record in them as well, you might be able to do even better in your ability to predict muscle activity?
So just for those of you who don't know all the anatomy that Andrew does, I'm talking about primary motor cortex. There are several premotor areas that are functionally somewhat distinct from each other. Some of them may have more kinematic information. Some of them have more force relationship. Some of them have to do with bilateral control. So there's a variety of information available throughout the cortex that we're not capturing at all. However, we're looking at the output to the spinal cord. So I think the signals that we're recording reflect already that other cortical activity. They reflect what the cerebellum is doing. They reflect what the basal ganglia are doing. They are all interconnected in the normal way. And we're just tapping off the output of that signal. Now, the other thing I should say is there are brainstem motor areas as well. They go to the spinal cord in parallel with the cortex. And in cats and rats, they're very important. In monkeys, they're kind of important. In humans, some of them remain rather important. Others have basically gone away. They're vestigial. The spinal cord itself also generates activity. So there's a lot of missing activity about the muscles. And yet we are able to compute VAFs under the good conditions of about 80% of relatively simple movements. So, but 60% of these things from the cage. So yeah, we're missing information for sure, but we're still able to capture a pretty good fraction of the muscle activity.
I mean, there are corticospinals from S1 and area 6 and stuff. So
absolutely true.
Yeah, so there could be some extra information.
Yeah, I don't deny that for a moment.
I'm curious about how stable the relationship is between cortical activity and muscle activity over time, or as a function of learning. or as a function of, say, surprise, like when you have those, anyway, just.
Learning is a great question. I'm going to tell you about someone else's experiment, which I think is wonderful. What I didn't tell you about this slide is, I told you about this slide. What I didn't show you is this. That was the projection 2 weeks later through the same UMAP projection. Not quite the same, but really very close. And we were pretty surprised by that. We've also looked over periods literally as long as 2 years, at which time our ability to make predictions from single neurons is completely gone if we use the same decoder that we did 10 years ago. On the other hand, if we compute a manifold on both day 0 and day 700, and then use canonical correlation or GANs, we've done it various ways, to align these two manifolds to each other, then we can continue to decode from that same fixed manifold decoder literally for 2 years. But that's at the manifold level, not at the single neuron level, because those are changing. Now, with respect to learning, I don't really have time to do this properly at all, but there was an experiment from University of Pittsburgh where they were very interested in this question and how learning was generated within the manifold or outside of the manifold. I'll go too fast for everybody to understand this, but basically they did a brain-computer interface in a monkey, asked them to make reaching movements, and then they perturbed this natural mapping, the intrinsic mapping, in 2 different ways. One, which allowed all the covariation between neurons to remain unchanged. Essentially, it's within the manifold. So the cursor, instead of going this way, all of a sudden goes off this way. The monkey can learn to adapt to that within a session, no problem. It's more or less like the motor learning that we would do to learn a new baseball bat or something like that. But if they introduced a perturbation that changed this intrinsic manifold or required the monkey to change it, it literally took weeks of coaching to get the monkey to learn that. So what I think is happening, and they think is happening, is that you're basically growing new connections in that 2 or 3 week period, or maybe strengthening connections, to essentially remodel this manifold, or maybe push it off in new directions. And that takes time, whereas the within manifold stuff is quick. And I think this has something to do with the question of why do we have 1 million neurons to begin with as well. We got a 1 million dimensional space to play around in. So we can learn all kinds of new manifold shapes. And we can relate sensory information to different kinds of sensory cues to different kinds of motor outputs, because we've got this incredibly flexible, extraordinarily high dimensional manifold on which to play.
Except that it's so stable and the covariance structure is so preserved over time.
Unless you need to learn a new behavior, which takes a few days or a few weeks. Then the question is, can I learn and Andrew has done some fascinating work on this years ago, actually. Can I learn 2 contexts? It may take a long time to learn the second one, but then switch between them easily. Probably that's the case. And the sparsity, you guys all know, the sparsity is part of what allows you to learn new stuff without screwing up the stuff you already know.
If there are no more questions.
Can I ask one more then? So does that mean learning, like to what extent is learning forging a new sort of unexplored dimension, or to what extent is it just using?
You tell me.
Okay.
Yeah.
So you don't have anything to say. You don't have thoughts about it?
I have thoughts.
All right, we'll talk another time.
Yeah, but that's a great question. It's fascinating. At some point, this begins to verge on philosophy because it's not clear we can even answer them, but they're certainly fun to talk about.
Let's thank Professor Lee again.
Thank you.